Good morning.
All right. Yes, Rob, good morning. Hey, thanks
Morning.
for joining us. Thanks all for joining us. I'm John
Absolutely.
with the Oshkosh Chamber, and thank you for joining us today on this little snowy morning in advance of the really frigid weather coming down here. So we're pretty excited today to have Rob Rigoni. It's Rigoni, right?
Rigoni. Yep. It's like
Uh,
rigatoni.
Local pharmacist here with Hometown Pharmacy. Been in town for a number of years. Ran into Rob, I don't know, a few weeks ago, and he was telling me about the vaccine. I was just fascinated by, by the chemistry behind that, biology behind that. And I thought, well, with this rolling out, a lot of people have a lot of questions about, the vaccine itself. There's a lot of stuff floating around out there, some real, some not so real. So we're going to explore that a little bit today. I mean, Rob's going to talk about the chemistry behind it, how this thing works, and we'll allow for questions at the end. So why don't, if anybody is listening in, if you have any questions, just use the Q&A tab on the bottom and ask those questions, and we'll review those at the end. We are recording this session, so it'll be archived on our website, and we'll push it out to others. And I think in the next several weeks, you'll see a lot more of these sorts of programs coming up. So we're pretty excited to be out there out in front with Rob on this. So with that, Rob, why don't you take it from here?
All right. Thanks, John. Thank you. Go ahead. We can move on. So the novel COVID vaccine, we can move on to the next slide. Just a little history of a vaccine. Evidence exists that the Chinese employed the first smallpox inoculation as early as 1000 AD. Edward Jenner, as we all know, is known for developing the first smallpox vaccine in 1776 using material from a cowpox pustule and then injecting its contents into its patients to give them immunity. Jenner discovered this because milkmaids had a resistance to smallpox. So interesting development there. They found out that women that were milking the cows somehow had a resistance to the smallpox, and it turns out because they had exposure to cowpox, which was very similar to smallpox. And he then extracted the contents from a pustule on a cow and injected it into his patients, and they therefore got immunity from it. Next slide. So let's go over some vaccine types. The live attenuated, meaning it's a live virus or bacteria, usually a virus, that has been weakened so that it can't produce a real severe infection, but a very mild infection, which allows our immune system to then mount a response and antibodies and immunity to it. Examples of that are measles, mumps, rubella, smallpox, chickenpox. Next slide, please. Types of vaccines. Another type is an inactivated vaccine. These are a killed version of the virus or bacteria. So basically, we take the virus, we kill it, so that then we inject it into the body, and we recognize these proteins, and then build an immune response from there. Examples are hepatitis A, influenza, polio, rabies. Important to note that inactivated vaccines don't provide immunity protection as strong as a live vaccine. That's why boosters are often needed. So that's important to remember. Next slide on that one. Types of vaccines. Another types of vaccines is the subunit recombinant polysaccharide or conjugate vaccines. These use specific pieces of the virus or bacteria, usually a protein or a sugar. Examples of these are hepatitis B, HPV, and pneumonia. These two often require booster shots. Next slide. Thank you. The other type is called a toxoid, where we use the toxin produced by the virus or bacteria. We take the toxin, inject it into our bodies. We build immunity to it via antibodies. And examples of the toxoid types vaccines are diphtheria and tetanus. And as you know, you need to get your tetanus booster every 10 years because these as well require booster shots. Next slide, please. So what's so new about the COVID-19 vaccine? Well, actually, it's not that new. The technology is not that new anyways. 30 years ago, scientists began exploring whether vaccines could be made much easier. Hence comes the mRNA vaccine technology. It is 30 years old, so it's not that new. We just haven't had too many opportunities to apply it. This COVID-19 virus has given them an opportunity to launch this new technology. Along the way, they learned that mRNA vaccines can actually generate a stronger type of immunity. Next slide, please. So this is a video, and I don't know if I can launch it. Oh yeah, there we go. Thank you. Thanks, Mark. So we can go ahead and play that, just kind of a good little summary on what's happening if it'll play.
Vaccines prepare the immune system, getting it ready to fight disease-causing organisms called pathogens. A vaccine is introduced to the body to mimic infection, triggering the body to produce antibodies against the pathogen, but without causing the illness. Conventional vaccines usually contain a weakened or inactivated pathogen, or a piece of a protein produced by the pathogen called an antigen. RNA vaccines are a new generation of vaccines. Instead of the antigen itself, RNA vaccines contain a messenger RNA, mRNA, that encodes for the antigen. Once inside the body's cells, the mRNA is translated into protein, the antigen, by the same process the cells use to make their own proteins. The antigen is then displayed on the cell surface where it is recognized by the immune system. From here, the sequence of events is similar to that of a conventional vaccine. Some RNA vaccines also contain additional mRNA coding for an enzyme, which, after being translated in host cells, can generate multiple copies of the antigen encoding mRNA. This essentially amplifies the production of antigen from a small amount of vaccine, making the vaccine more effective. These are called self-amplifying RNA vaccines. RNA vaccines are easier and safer to produce than conventional vaccines. This is because mRNA molecules can be synthesized in a cell-free system using a DNA template with a sequence of the pathogen. While conventional vaccines usually require a more complicated and risk-prone process of growing large amounts of infectious pathogens in chicken eggs or other mammalian cells. Without the risks of being contaminated by infectious elements or allergens from egg cultures, RNA vaccines are also safer for patients. Because protein synthesis occurs in the cytoplasm, RNA molecules do not need to enter the nucleus, so the possibility of them integrating into the host cell genome is low. RNA strands are usually degraded by cellular enzymes once the protein is made. The relative simplicity of the production process makes it easier to standardize and scale, enabling rapid responses to emerging pandemics. Other advantages include lower production costs and the ease of tweaking RNA sequences to adapt to rapidly mutating pathogens. On the minus side, it can be challenging to deliver mRNA effectively to the cells, since RNA sequences and secondary structures may be recognized and destroyed by the innate immune system as soon as they are administered intravenously. These limitations can be overcome by optimizing codons, using modified nucleosides to avoid recognition, and packaging RNA into protective nanoparticles. Another disadvantage is that most RNA vaccines require uninterrupted refrigeration for transportation and storage, which can be a hurdle for vaccine distribution. Research is ongoing to engineer thermostable vaccines.
Oh, thank you. Thanks, Mark. Okay, so basically just a quick synopsis of that video. The novel coronavirus is studied in labs, so they were able to sequence the virus, knowing its DNA makeup, carve out a piece of the DNA that produces the spike protein. So then once they knew what that spike protein was, then they could create the mRNA to then inject into our cells to produce the spike protein from our cells. And I'll explain that here briefly. So they sequence the code, the mRNA that codes for the spike protein is purified and injected, body produces the spike protein, the immune system produces the antibody. So think of this, so the mRNA is the recipe card, and I'm probably dating myself. You know, you got your Aunt Margaret's favorite fruitcake recipe, and you're bringing, instead of making it at home, you're bringing the card over to your friend's house and you're using their equipment to make the fruitcake. And that's basically what this is doing. We are taking the DNA structure for the spike protein, building an mRNA, which stands for messenger RNA. The messenger RNA is then injected into our arm, or for the injection, and then the cells will take it up because the mRNA is coated in a lipid. It's what they call a lipid nanoparticle, and so that mRNA will get absorbed by the cell, the surrounding cells, wherever we inject. Usually, it's injected in your arm. So the surrounding cells will then take up the mRNA. The mRNA will find a ribosome, and these ribosomes are in our cytoplasm. These are like little machines, and they'll grab the mRNA and they will start to reproduce the spike protein. And that spike protein is what the virus uses to attack us and get into our cells. So that spike protein will then migrate to the cell wall, and then an immune cell, like what we call these macrophages, will come and they will swallow that cell in a process called phagocytosis. It's a phagocyte. And so what that does then, it will take that cell that it engulfed, take it up to the lymph nodes, and the lymph nodes will then start to produce antibodies through our B cells. So we can go on to the next slide and I'll kind of go over that real quick again. So basically this is just kind of redundant in what I was saying, but here's the spike protein. We produce the mRNA sequence. We put a lipid coating in it because our cells are loaded with what they call phospholipid barrier around our cells, and they are very attracted to fats. So it's called lipophilic. So we put that lipid coating on the mRNA in order for the cell to take it up. And then, like I said, we get it in the cell, get it in the cytoplasm. The ribosomes get to work. They start producing the spike protein. Spike protein then migrates to the cell wall. Our immune system takes over. Macrophages come in, swallow it up, and then take it up to the lymph nodes where we find the T cells and we find the B cells, and antibodies are produced, and the T cells are motivated to go out there and start killing every cell that has that spike protein on it. Next slide, please. Okay, and this is kind of redundant, but the spike protein coated in the mRNA is encapsulated in a lipid nanoparticle. The nanoparticle is taken up by the cell. mRNA goes to the ribosome where the protein synthesis takes place. Spike protein is made. Once made, the spike protein migrates to the surface where the immune system will then mount and attack an immune response. Next slide, please. Once vaccinated, a very specific and significant antibody response is created. Why is this important? Compared to traditional vaccines, mRNA vaccines can actually generate a stronger type of immunity by stimulating the immune system to not only make antibodies, but also to stimulate killer cells, the CD4 and CD8, a double strike for you Cobra Kai fans. It's a double strike. So we're not only producing antibodies, we're actually stimulating our killer cells to go and seek out cells that may have the spike protein on it. This immune response from the vaccine persisted for at least 3 months, likely more, after the second dose, and remains significantly higher than that seen in individuals who have actually had COVID. Vaccine efficacy is around 95%. And this is just speculation, but about 5% may be at risk for contracting COVID, but the illness will be milder because of some immunity. This is speculation. We don't know for sure, but similar to our influenza vaccines, even when they try to figure out which strains are coming our way and we immunize, even if we do if they get it wrong, we still contact influenza, we may get a milder case because we do mount some immunity to it. So we're hopeful this is the same process. Next slide, please. So just kind of looking at the math and how we calculated the efficacy of the Pfizer vaccine. And this is the Pfizer chart. They had 43,000 people in their study. Of the placebo group, they had 162 infected with an infection rate of 0.74%. It doesn't sound like a lot, but when you spread that across the population in the US, it's about 2.5 million. With the vaccine, they had 8 people infected out of the 21,830 for an infection risk of 0.04. Quite significantly lower, a 0.7% difference. Now, if you divide that 0.7 divided by the 0.74, that's where we get that 95% efficacy rate. Next slide, please. So what groups are currently authorized to get the vaccine? I think currently now, this slide's a little older. We are in 1B, March 1st. They will even expand who's eligible for it even further. Follow the Winner County website for current authorization as to who is recommended to get this at this time. Under the FDA's emergency use authorization, the following age groups are authorized to receive the vaccine. For the Pfizer, BioNTech, ages are 16 and older. For Moderna, it's only 18 and older. I just put a little asterisk here. If pregnant people are part of a recommended group to get the vaccine, example, a healthcare worker, they can get a vaccine. And the thought process is, if the benefits outweigh the risks, go ahead and get vaccinated. The vaccine is administered intramuscularly. It could be a shot in your shoulder. The Pfizer is a 2-dose series with 3 weeks apart from the first to the second dose. The Moderna is also a 2-week series or 2-dose series with 4 weeks apart from first to second dose. The vaccines are not interchangeable. Next slide, please. Side effects, most common, pain and swelling at injection site, which is common for all these vaccines. We poke you in the arm with a needle, so you're going to have some soreness. Fatigue, headache, myalgia, which is muscle pain, muscle aches. This is an expected side effect of vaccinations and more likely after the second dose of the vaccine. People that I've talked to, it seems that the second dose, they get this fatigue, headache, myalgia, maybe a slight fever, and that's your immune response. That's your immune system responding to that second dose, and it's mounting a immune response. So that's a good sign. Recent CDC reports show 11.1 cases of allergic reaction per 1 million. People with 71% occurring within 15 minutes of vaccination. So if you've never had any past history of allergic reactions to vaccines, once we immunize you, we'll ask that you sit for 15 minutes to make sure you don't have any reactions. If you've had a history of allergic reaction to vaccines in the past, we will ask you to stay for about 30 minutes to observe you to make sure you don't have any allergic reactions. Next slide, please. So I thought I'd put a new slide in here about the Johnson & Johnson vaccine and kind of go over some info in regards to that. The Johnson & Johnson vaccine is not an mRNA vaccine. It is a genetically altered adenovirus, which is like our common cold, that produces our spike protein. The virus is then denatured so that it cannot replicate itself or expand in numbers, but it can still interact with our cells, and the DNA from the virus enters our nucleus, where then the mRNA is produced, sent back out to the ribosomes, the protein-making machines. They will make the spike protein, and then our immune system will mount an attack to form immunity. So a little different. So they actually take an actual virus, mutate it, genetically engineer it, and inject it into us. It's nice thing is a single dose vaccine. So it requires 1 dose, which is nice as far as compliance is concerned. However, it only has a 66% overall efficacy, 72% in the US. This has to do with the studies in Europe, Latin America, and South Africa, where the prevalence of the variants was more common versus that in the US. What was interesting, though, is after day 28 from the inoculation, it had an 85% efficacy overall in preventing severe disease and preventing hospitalizations and death. Another pro of the Johnson & Johnson is the vaccine cold chain requirements are less complicated and burdensome compared to the Moderna and the Pfizer vaccine. The vaccine can be stored in the refrigerator for up to 3 months. Next slide, please. Dispelling the myths. There was a lot of myths about sterility. There has been no safety concerns identified in development of sterility in the reproductive toxicology study. There's no egg allergy concerns. It does not change our DNA sequencing. There is no mercury, and there is no 5G chip, so the government won't be tracking you or controlling you. Next slide, please. So the common questions are, should I get the vaccine if you've already had COVID? Absolutely. They've done the studies. You'll have antibodies after you've contacted COVID, but they found that people who've been vaccinated have significantly higher amounts of antibodies versus the convalescent serum, which is someone who's had COVID. So definitely get the vaccine. Your immune system will be stronger. The amount of antibodies that'll be circulating in your system will be higher, and you'll be better protected. The other question is, how did they mass produce the vaccine so quickly? Well, as you've seen in the video, it's basically they were able to produce the mRNA in a lab without having to grow a virus in an egg and then then killing the virus, then having to purify it and try to extract all the egg protein out of it. It's quite a lengthy process and a cumbersome one. This is all done in the lab, so they can crank this thing out pretty quickly. They actually had vaccine made and ready once the FDA gave them approval, waiting for them to give the approval. So that's why they were able to get so much vaccine out so quickly. What are the differences between the Pfizer and Moderna? Not much, same technology. Moderna's cold chain is less cumbersome. The Moderna can be kept in the refrigerator for up to 30 days, where the Pfizer can only be kept in the refrigerator for 5 days. Moderna has some proprietary stabilizers that they're using in their vaccine that allows it to be more stable over a longer period of time. Next slide, please. That's it, I guess. Thank you very much for calling in this morning and looking at this PowerPoint presentation with me. And if there's any questions, let me know. Thank you.
Yeah, thanks, Rob. Very, very informative. So if anyone's listening and has a question, just use that Q&A button on the bottom, and we'll get to it. I've jotted down a few things that I thought people might be interested in. I am, and I think you've addressed some of those. But a lot of uncertainty out there, as you know. And even though the science is there, people are still like, well, this is still new. It's new. This whole COVID-19 thing's new to everybody. But you talked about mRNA, mRNs, DNA. One of the questions that keeps coming up is, will the COVID-19 vaccine affect my DNA? I mean, how does that?
No.
People talk about, oh my God, my DNA is
yeah. So a couple of things. So if you look at the video was explaining, that mRNA molecule is just basically a recipe, and it has the DNA sequence for how the spike protein is made. Goes in the cytoplasm, and there's these little ribosomes, which are little machines that will then produce the spike protein. That spike protein never enters the nucleus. The mRNA never enters the nucleus. It goes up to the surface of the cell, and then our immune system comes in. There's like a macrophages. These are our first line of defense, basically, in this immune system process. And what the macrophage does is it says, hey, that looks strange to me. What is that? And then it will go in and it will engulf that cell through a process called phagocytosis, eats that cell, then carries that cell into the lymph system where our T cells are, where our B cells are, and then mounts an attack. So the cell actually gets destroyed by our immune system. And then with the Johnson & Johnson one, the virus actually does get into the nucleus, and the virus will then, just like all viruses, then will have the nucleus and produce its own genetic material through the ribosomes. Then again, that cell is destroyed. It's killed by our immune system. So it will not mess up with our DNA because these cells are destroyed by the immune system. Especially, it's not an issue with the Moderna or the Pfizer one. In theory, with the viral one coming from Johnson & Johnson, that's basically how a virus works, but those cells are destroyed either by the virus or they're destroyed by our immune system.
Okay. Well, we're getting some questions in from those listening in. First question is, why has it been so difficult to get the vaccine?
Well, what they're trying to do is they're trying to identify groups that are the highest risk. Obviously, it's your frontline healthcare workers, your first responders. So they want to make sure they get the people that are on the front lines getting the vaccine first. And then now in Wisconsin, we are vaccinating those 65 and older. Next phase is coming in March. So it's an effort to try to identify the highest risk groups and then make sure we get them vaccinated first. But with that, it becomes cumbersome. Unlike other vaccines where it's like, hey, come get vaccinated, first come, first serve. Similar to, so they're trying to control it, and it becomes very cumbersome, unfortunately. But hopefully over time, as more of these vaccines come online, we're going to be able to start immunizing people at a much faster rate.
Well, other than the Johnson and Johnson vaccine, the other two require two doses. The question they have is there a difference between the first and second doses, or are they the same vaccine?
So it's the same. So right now we can't interchange. If you get the Pfizer first time, you need to get the Pfizer the second time. Same with the Moderna. Moderna the first time, Moderna the second time. Right now they don't recommend interchanging them. I guess if they had to for emergency reasons, I guess at some point they may say, yeah, go ahead and do it. But as of now, get the Pfizer first, you get the Pfizer second. The dose is the same. For Pfizer, you get 0.3 mLs. For the Moderna, you get 0.5 mLs. The first dose, usually we don't have much of a response. You get a sore arm. But a lot of people on that second dose, they start getting the body aches, mild fevers. They just feel terrible for a day or two because now your immune system is like, hey, I know what that is. I need to mount a response to it. And that's a good sign. That means your immune system is working. And we're seeing those side effects more common in people 50 and younger because we know that people 50 and younger, our immune systems are stronger. In the population of 65 and older, they don't seem to get as many side effects on that second dose compared to the younger group, is because their immune systems aren't as strong. And this is why we have a high dose influenza vaccine for those 65 and older, because we're trying to get a good immune response. And we know as we age, our immune systems get weaker and weaker, and they don't quite respond as well as we'd like them to.
Here's a question that came in. Can you explain the egg thing? Wait,
Yeah.
so
So
other vaccines as opposed to mRNA.
okay.
mRNA. There we go.
So we've been using chicken eggs to produce our influenza vaccine for, I don't know, for probably 50 years at least. So actually, when I was in pharmacy school, I got the opportunity to go to Park Davis, and they had a factory where they had all these eggs, and they were growing influenza virus inside these eggs. So in order to grow the influenza virus, so like right now, our process for the influenza vaccine flu shot is the CDC, the WHO, they try to figure out what strains are coming. Usually they try to figure out what strains are coming out of Australia mostly and parts of Asia. And they say, okay, I think this is going to be this strain of influenza coming our way. They then identify that, then they grow it in eggs in mass amounts. They grow the virus in eggs in mass amounts, and then they have to kill the virus and then strain out all the egg protein and get as many impurities out of it as possible. The downside is it takes a lot of time. It's a lot of effort. It's very crude technology compared to the new technology today. And then there's also the risk for egg allergies. If a person has an egg allergy, then they're going to have an issue with this vaccine because it comes from eggs.
Will people have to have this vaccine every year, or is it two and done?
Well, right now it's two and done. It's two and done. And I know there's a lot of media reports out there saying, well, maybe we need to get a COVID shot every year, similar to influenza shot. I mean, as of now, no. I mean, there are 2 or 3 variant strains. And there was an article, I think it was in JAMA, maybe it was a week or so ago, even the Pfizer and the Moderna, if the person was to contact this new strain, this variant, they're still going to have about 70% efficacy with that. So biggest key is we got to get to that herd immunity, which is probably like 70 to 80% of the population. We can get the herd immunity, then we can protect the more vulnerable in our population. And that's the whole point of trying to get everybody vaccinated, is to protect the vulnerable in our population.
I think getting these vaccines administered has been difficult for folks. What happens if you have the first dose but you don't get the second dose? You have to start over, or?
No, get it as soon as you can. There is no, you do not have to restart the series. So, I mean, obviously with Pfizer, it's a 3-week window is recommendation. Moderna is a 4-week window. But if for some reason they miss it or they can't get it or whatever, they get it as soon as possible. There's no need to repeat the series.
Will Hometown Pharmacy vaccinate individuals who are associated with one of the major health organizations like Theta Care or Aurora?
So we've vaccinated some healthcare workers. Most of the time, those individuals that are working for the large groups, they've been vaccinated already. But independent optometrists or dentists or other pharmacists in the area that want to get vaccinated, they can get on our wait list, and they will be prioritized because they are part of that 1A group.
Question came in, will the flu vaccines become, my eyesight's awful these days. It's mRNA. Yeah, there you go.
I hope so. I mean, there may be a motivation and incentive now to do that, but it takes time because it's such the way they've been making influenza vaccine is it's so integrated. It's part of the process. These companies have to now scrap that process entirely and start producing influenza with mRNA technology. So I hope so. I would not be surprised if we see an mRNA influenza vaccine in our near future.
We've been hearing a lot of stuff from the health departments, CDC. People are getting vaccinated, but they're still telling people to be diligent. Still keep masking up, being social distancing. Why is that? I mean, if you had the vaccine, why do you need to be still that cautious?
What was nice is now the CDC came out and said once you finish your 2-shot series, and it takes about 2 weeks to mount the perfect immune response. So you get your first shot. There was one study, I think Pfizer did the study and reported the data, but Moderna did not. After your first shot, 2 weeks out, they figure you got about a 52% efficacy just after one shot. And then after you get your second shot, 2 weeks out from that, that's when we reach that 95% efficacy. Now the CDC just came out, says once you finish your 2-shot series, you no longer have to quarantine any longer if you had contact with somebody. To answer your question, I think it's because we got these vaccines in place in less than a year. We don't have any long-term data as far as once you're vaccinated, can you still transmit the virus? We don't know. But if you have full immunity, it seems like that would be unlikely. But say you're the 5 percentile that still contacted a mild case of it, case of the COVID, then there's still a good chance that you could transmit it. So they don't want people to be overconfident and say, well, I've been vaccinated, now I don't have to wear a mask. But if you happen to be that 5% who could still have a low grade infection, there's obviously a risk that you could be transmitting it. And we may see in the future that once we get to this herd immunity, is that maybe it's not a bad idea if you get sick and you've got symptoms, simply put a mask on to protect others. But as of right now, until we get all the data, we don't know if the vaccine actually keeps us from spreading it to somebody else. You would think in theory that it would, but again, we got that 5%. Now with these new strains, if we drop down to that 70%, that means there's a chance that 30% of the people could still have a mild illness that could spread it. So I hope that answers the question.
So once you're vaccinated, how long is that vaccine effective? I mean, will you have to be, get a revaccination at some point?
Well, so the studies that Moderna and Pfizer did, they looked at 90 days out. And so there's a lot of data coming back now. Some data saying they expect it to be longer than 90 days, but we just need to collect it. We just don't have the data, so you can't really say for certain. But after 90 days, the antibody count in our blood was significant, very significant after 90 days. So we know after 90 days of immunization that you've got a full-blown immunity. But as we study this vaccine more, hopefully we'll get more data, and hopefully it lasts just like a lot of vaccines. Maybe we get lucky and it lasts as long as a tetanus vaccine and it's 10 years, or measles or polio and it lasts your lifetime.
Right. The younger demographic out there hasn't had as isn't been as vulnerable in terms of the effect of COVID-19. They've been getting it, but I know my daughter and some of her friends, they've their friends have gotten it. It's been the sniffles and that sort of thing.
Mhm.
Is it advisable for them to get vaccinated, or should they just ride it out and
Yeah.
see what happens?
So, I mean, the infection fatality rate in kids 18 and younger is like 0.003%, and it's very rare. Interesting, 81% of those infected have mild symptoms. Um, so, in that group right now, the Pfizer is 16 and older indicated. The Moderna is 18 and older. So the kids will not be vaccinated at this time, and they are the least vulnerable, like you said. But those that age group that's 18 to 40, and I think their fatality rate is like 0.05, if I remember off the top of my head. It's still very low. Um, we want them immunized because they are going to provide that herd immunity. They're going to be able to protect the most vulnerable in our society. And that's why we get them immunized. We don't get them immunized because we're so worried that they're going to have bad outcomes from the virus. We want them to be immunized because they are going to protect the most vulnerable in our society. And that's why we encourage them to get immunized. I mean, 0.05%, you spread that out over that population 18 to 40. I mean, there's still a good number who are going to have bad outcomes when you spread it across, 50 to 70 million people. But by getting them immunized, they're going to provide that herd immunity to protect the vulnerable parts of our population. Grandpa and grandma, when they go visit grandpa and grandma for 4th of July parties and Christmas and that, they're doing their part to protect grandpa and
Question that came in is, so is this truly a vaccine, or is it really a treatment if you can still be a carrier?
grandma. Well, it's a vaccine. The problem is we don't have the data to say conclusively say, hey, we know once you get vaccinated, you cannot transmit this virus. But if you're part of that 95% that is able to mount a true immunity at 95%, in theory, you shouldn't be transmitting the virus. But if you happen to be that 5% who still comes down with a mild case of it or a moderate case, then there's still a chance that you could transmit it. But right now, we just don't have enough data to say one way or the other. But it is truly a vaccine.
Um, we talked about reproduction and younger folks. If the vaccine hasn't been around long enough, for the data to be collected, how can we be, certain that it doesn't affect the reproduction systems of young females?
Well, they did the toxicology study, the sterility study. They didn't find anything in there. There was nothing there as of now. Obviously, as this thing goes out, years out, anything can happen at post marketing, post launch. But the thing of it is that it's basically, if you look at the science behind it, it's basically the same process as our other vaccines. Instead of taking the virus's actual protein or sugar, we're actually making it in the lab. We're just replicating it in a lab and then giving it to the individual. So, we don't know the safety data long term. We're still collecting data on it, but the preliminary studies that they did, they showed no risks for sterility.
It looks like we're winding down on questions, but one other question here that came in: what percentage of the population do we need to have been vaccinated so we can reach the elusive herd immunity?
I've seen different numbers, but the most common I'm seeing is 70 to 80%, 70 to 80%
Yes,
vaccinated.
that's good.
So that's
Well,
a lot of people.
yeah, that is a lot of people. Now, would that include also folks who have already had
Yes,
COVID-19? Okay.
yes. Well, so there's interesting stat about vaccinations. We have never hit herd immunity without immunizations. So that's part of it. So you have the folks that had prior COVID and you have the folks that have been immunized. That's going to get us to herd immunity. But we want those people who've had a history of COVID infection to get immunized too, because they're going to have even a stronger immune response, and they're going to be less likely to transmit it to somebody else.
Sounds good. Well, Rob, we really appreciate you joining us today. It's fascinating.
Thank you.
We'll
Thank you.
be pushing this out to probably to the membership. It's been recorded, so if those are listening now can go back and listen again and tell others. I think this is something that has been on a lot of people's minds. Question did come in about whether we can share the PowerPoint with attendees, and we will push that out to those that registered today. So you'll be looking for those. So again, we thank you for joining us. Rob, thank you for joining us. Good stuff. And as always, we keep these Zoom webcasts coming. So if anybody has any ideas on other topics we should do, we're always looking for new topics and new presenters. So with that, we appreciate y'all joining us, and we will see you again very soon. Thank you
Thank you,
again.
everybody. Thank you. Have a good weekend.
You too.